Why do our bodies age and die? And can science extend healthy human life without creating new biological risks?
It is, I mean, it's the ultimate question. But to really answer it, we have to look past the headlines, the ones about tech billionaires trying to buy immortality.
Right, the Silicon Valley quest to live forever.
Exactly. Because yes, aging research has successfully identified these interconnected molecular and cellular processes, and we can actually modify them in laboratory organisms. We can change the biology of a roundworm or a fruit fly.
Oh, wow.
Yeah, but here's the massive caveat. Slowing a biomarker in a test tube or, extending a mouse's lifespan in a completely controlled, sterile lab setting, that absolutely does not prove that an intervention will safely extend human health span or lifespan.
Because we're not mice in sterile bubbles.
Precisely. The jump from a localized cell culture to a biologically complex human being involves massive and often competing biological risks.
Yeah, the gap between a headline and human biology is just massive.
Yeah.
So for this deep dive, we are exploring Venki Ramakrishnan's book, Why We Die, The New Science of Aging and the Quest for Immortality. And to really understand what's happening inside us, we're going to trace the entire story of human aging.
It's quite a journey.
It really is. We'll start by asking why evolution built us to decline in the first place, and then zoom right into the cellular engine room to look at DNA damage, telomeres, and epigenetics.
Right, the mechanics of it.
Yeah. We'll explore how our ribosomes manage protein quality, how our mitochondria generate energy, and how nutrient sensing and autophagy act as the body's, recycling centers.
And what happens when they fail?
Exactly. Which leads to cellular senescence, stem cell exhaustion, inflammation, and declining immunity. We'll even look at the animal kingdom to see if comparative longevity holds any secrets before we, strip away the hype to evaluate biological age measurements, calorie restriction, and those experimental drugs everyone talks about.
The pharmacopoeia, yeah.
Right. And we'll end by looking at what actually constitutes healthy aging today and the profound ethical questions a longer life would force upon society.
But before we delve into all the mechanics of those processes, I do need to provide a clear safety statement. This discussion is educational and is not medical advice.
Very important.
Yes. We do not recommend fasting, calorie restriction, supplements, hormones, prescription medicines, off-label drugs, or any experimental treatments. Any personal medical decisions really should be made directly with qualified healthcare professionals who understand your specific medical history.
Perfect. Okay, so let's start at the very beginning. Not with how our cells break down, but why evolution allowed aging to exist at all.
The big why.
Right. Because I think a lot of people assume that nature would want us to live forever, right? So we could just keep having offspring and passing on our genes.
It seems logical.
It does. But natural selection actually weakens with age. Traits are only favored if they help an organism survive and reproduce early in life. Once an organism has passed its reproductive window and, raised its young to independence, the evolutionary pressure basically drops to zero.
We call that the selective shadow. Evolutionary biologists use that term because the forces of evolution are completely blind to whatever happens to you after you've reproduced.
The selective shadow, that's a great term. Which brings up the first major theory we really need to explore, which is the disposable soma theory.
Right.
And soma just means the body.
Exactly.
So the idea is that an organism only has a finite amount of energy available from its environment. It has to make a budget.
It's all about resource allocation.
Right. Does it spend that energy on growth and reproduction, or does it spend it on perfect, eternal cellular maintenance? And evolution always prioritizes reproduction. The body is just treated as a disposable vehicle to pass on the genetic code.
It's a harsh reality.
It is. I kind of think of the human body like a Formula One race car.
Oh, I like that.
Yeah, because the engineers built it to do one thing: drive incredibly fast, survive a very specific aggressive race, and cross the finish line. It was never designed with the intention of commuting back and forth to the grocery store indefinitely for 80 years.
No, it would break down immediately.
Right. The parts are going to wear out because they were never built for eternity.
The race car analogy is really useful for understanding that energy allocation, but we actually have to layer another concept on top of it to see the full biological picture.
Okay, what's that?
That concept is antagonistic pleiotropy.
Antagonistic pleiotropy. Okay, so pleiotropy being a gene that controls more than one trait.
Correct. So antagonistic pleiotropy suggests that a biological pathway or a gene variant that provides a massive benefit for early growth, development, or immunity.
Like when you're young.
Exactly. That same pathway can actively contribute to our structural decline later on in life.
Wait, really? So nature actively programmed things that harm us later?
Well, evolution didn't program a specific death switch to eliminate us. That's a common misconception. It simply favored aggressive, early survival mechanisms.
I see.
Let's ground this in how we evaluate evidence. When we look at observational studies of these pathways, we really have to look closely at the trade-offs. For example, a robust cellular growth pathway helps a young mammal build strong bones. It helps it heal quickly from injuries, ensuring it survives long enough to reproduce.
Right. You want rapid growth when you're a kid.
Exactly. But that exact same robust growth pathway, if it's left active into late life, becomes a primary driver of cancer and cellular exhaustion.
Oh, wow. So if researchers try to, modify these pathways in the lab to suppress that late life decline, they're going to run straight into those evolutionary trade-offs.
Always. You can't avoid them. When you see a claim that an intervention suppresses the late life aging pathway, you must ask, what clinical endpoint was measured beyond simply aging?
Like, what else happened to the animal?
Exactly. Did they inadvertently impair that organism's ability to heal a wound? Did they compromise its ability to mount an immune response against an infection?
Right, because they turned down that pathway.
Yes. The label anti-aging implies a single directional benefit, but biologically, we are almost always trading one risk for another.
Trading one risk for another. Okay, let's follow that idea of structural decline down into the actual biology.
Let's go to the cellular level.
Yeah, the cellular engine room. Now, Venki Ramakrishnan won a Nobel Prize for his work on ribosomes, so it makes total sense that his book heavily focuses on the translation of genetic information.
It's his specialty.
Right. So our DNA is essentially the instruction manual. That manual is transcribed into RNA, and then those instructions are read by the ribosomes, which manufacture proteins based on that code.
And proteins do the real work.
Exactly. Proteins do almost everything in our cells. They're the structural building blocks, the enzymes, the communication molecules. But this system is not flawless, is it?
Far from it. The fundamental driver of the aging process at a molecular level is the accumulation of errors.
Because the environment damages the DNA over time.
Right.
Right, like UV radiation, chemical exposure, simple metabolic stress.
It is, but it's not just environmental damage. Simply existing and functioning creates errors.
Oh, really? Existing.
Yes. Even if an organism were kept in a perfect, sterile environment, the DNA replication machinery just makes mistakes. The RNA transcription process makes mistakes. The ribosomes themselves occasionally mistranslate the code, which results in misfolded proteins.
And going back to the disposable soma theory, the energy cost of maintaining a 100% perfect proofreading and repair system would just be too high.
It would be impossible to budget for.
Right, so the body just accepts a certain margin of error. But as those errors in the DNA and the proteins accumulate decade after decade, they start to disrupt cellular membranes, enzymes, and the way our cells signal to one another.
Which brings us to the most, I'd say, popularized aspect of cellular aging.
The one everyone talks about.
Yes. Because when people talk about cellular decline, they rarely discuss ribosomes. They almost exclusively want to talk about chromosomes.
The telomeres. This is the concept everyone seems to know, but let's make sure we understand the actual mechanics here.
Let's define them.
Yeah, so telomeres are the protective caps of repetitive DNA at the very ends of our chromosomes. I think of them like a prepaid Metro card for the subway.
That's a good one.
Right.
Yeah.
Every time a cell divides, it has to swipe the card, and a little bit of the telomere sequence is lost. And when the card's balance hits zero, when the telomere becomes too short, the cell is basically locked out of the turnstile. It is physically incapable of dividing anymore.
I am so glad you avoided the common shoelace analogy because the Metro card illustrates the functional limit perfectly.
The shoelace is a bit played out anyway.
It really is. However, this popular awareness has led to a massive, highly commercialized misconception. We need to state clearly that telomere length alone does not define a person's biological age.
But wait, if short telomeres are the reason our tissues stop refreshing themselves, shouldn't telomere length be the ultimate test of how old our bodies are?
No, because aging is an interacting set of changes across multiple tissues. Telomere length is incredibly highly variable. It differs between different organs in the same person, it differs between different cells in the same organ, and it even fluctuates based on temporary immune responses.
Wait, really? It can fluctuate.
Yes. So a single blood test measuring telomere length is a deeply incomplete indicator of whole body aging. It is just one small piece of a much larger puzzle.
Okay, even if it's just one piece, let's play devil's advocate for a second.
Go for it.
If the cell stops dividing because the telomere gets too short, and that contributes to tissue aging, why don't we just find a way to add value back to the metrocard?
Ah, the telomerase question.
Right, if we use an enzyme like telomerase to artificially lengthen the telomeres, wouldn't the cells just keep dividing and refreshing the tissue forever?
This is where we have to critically test the evidence. What actually happens when scientists artificially extend telomeres in animal models, the data reveals a very, very grim biological reality.
It is.
Strategies that promote continuous, limitless cell division to preserve tissues also enable malignant growth.
You're talking about cancer.
Yes. Limiting cell division, forcing the cell to stop swiping the metrocard, is actually one of the primary ways your body stops a damaged cell from multiplying out of control.
Oh, man.
If a cell has accumulated DNA damage, you do not want it to have an infinite capacity to divide.
Oh, wow. So short telomeres aren't necessarily a design flaw causing us to age. They are a defense mechanism keeping us alive long enough to reproduce.
Exactly.
So if we force the telomeres to stay long, we might theoretically prevent tissue aging, but we drastically increase the chance that the organism just dies of a massive tumor.
Exactly the trade-off I mentioned earlier. Any longevity intervention targeting the cellular engine room has to perfectly balance tissue renewal with tumor suppression. There is simply no biological free lunch.
No biological free lunch. I love that. Okay, so DNA and telomeres are essentially the hardware of the cell. But what happens to the software over time? We need to talk about epigenetics.
Yes, the software.
This is fascinating to me because every single cell in your body, whether it's, a skin cell, a liver cell, or a neuron, it contains the exact same DNA, the exact same instruction manual.
Every single one.
So how does a neuron know to act like a neuron and not a liver cell? It comes down to epigenetics.
The chemical markers that sit on top of the DNA.
Exactly. Epigenetics involves chemical marks like methylation and the physical structure of the chromatin, which basically dictate which chapters of the instruction manual are open and which are glued shut. The skin cell has the liver genes turned off.
Right.
But as we age, we experience something called epigenetic drift. To use another analogy, if the DNA is the keyboard of a piano, the epigenetics are the sheet music telling the cell which song to play.
I like the sheet music analogy.
Thanks. But over time, due to environmental stress, chemical exposure, and just the sheer mechanical friction of cell division, it's like someone is spilling coffee on the sheet music.
The notes get blurred.
Exactly. The keys still work fine, but the cell starts playing the wrong notes. Genes that should be permanently suppressed get accidentally turned on, and genes that should be active get muted.
And this molecular drift is measurable. And that measurability has spawned an entire industry of biological age testing.
The epigenetic clocks. You can mail in a cheek swab, and a company will look at the methylation patterns on your DNA and tell you that even though you are chronologically 45 years old, your biological age is 38 or 52.
Which brings us to another critical misconception that requires immediate correction.
Lay it on me.
Aging is not controlled by one single centralized clock ticking away in our biology. There are multiple epigenetic clocks, and they measure completely different sets of methylation sites.
So there isn't just one master clock.
Exactly. And more importantly, a younger biomarker result on one of these commercial tests does not automatically mean better health or a longer maximum lifespan.
I think of the epigenetic clock tests kind of like the check engine light on a car's dashboard.
How so?
Well, the light is a signal that something is happening under the hood. But resetting the computer so the light turns off doesn't mean you actually fix the engine.
That is a highly accurate way to look at it. When we evaluate the hype around epigenetic clocks, we have to ask, is the clock merely a surrogate marker?
Right.
If a person changes their diet or takes a supplement and their epigenetic clock score drops by 5 years, did their physical function actually improve? Did their disease resistance increase? Or did they just change the oil light without changing the oil?
Right, because moving a surrogate biomarker on a spreadsheet does not mean an organism will actually survive longer in reality.
Precisely.
But scientists are trying to actually fix the engine, right? Let's discuss partial cellular reprogramming. Because researchers have figured out how to use specific transcription factors, the Yamanaka factors, to strip away those coffee stains on the sheet music.
They have.
They can take an adult cell and reprogram its epigenetics back to a more youthful state.
Yes, in localized tissues, in laboratory animals, this has shown some remarkable localized rejuvenation. But we must categorize systemic partial cellular reprogramming in humans strictly as promising but unproven, and currently highly speculative.
Why speculative? If we can restore the youthful software, wouldn't that solve epigenetic drift?
Because resetting a cell's identity carries severe risks. We have to ask what happens to the whole organism, not just a tissue sample in a petri dish.
Okay.
If you push the reprogramming too far, the cell forgets its identity completely. A heart cell forgets it is supposed to be a heart cell.
Well, that sounds bad.
It is. When cells lose their designated identity and begin to revert to a stem cell-like state in a living adult organism, it often results in teratomas, massive, chaotic tumors containing multiple tissue types. You'll have hair and teeth growing where they shouldn't. Systemic reprogramming in humans is not a near-term reality. The risk of abnormal fatal growth is simply too high at this stage.
Wow. Okay, so resetting the software is incredibly dangerous because the cell might forget what its job is.
Exactly.
That brings us logically to how cells manage the daily damage of doing those jobs. Every day, the cells have to clean up the microscopic garbage they create. This involves two main processes, proteostasis and autophagy.
The cleanup crews.
Right. Proteostasis is protein quality control. The cell has these chaperone molecules that make sure proteins are folded into the correct 3D shapes, because a misshapen protein is useless or even toxic.
And autophagy.
Autophagy is the cell's internal recycling program. When components get damaged, the cell creates a little garbage bag, swallows the broken parts, and breaks them down for spare parts.
But as we just established with epigenetic drift and accumulated DNA errors, the efficiency of these systems declines over time. The chaperones fail, the recycling center gets overwhelmed.
And the garbage starts to pile up inside the cell. And nowhere is this more critical than in our mitochondria, the cellular power plants.
A vital piece of the puzzle.
Mitochondria take the food we eat and the oxygen we breathe, and they generate ATP, which is the chemical energy of the body. But running a power plant creates exhaust. In this case, the exhaust takes the form of reactive oxygen species, or ROS.
Yes.
These are highly unstable oxygen molecules that can damage surrounding cellular structures.
If we look at the historical context of aging research right here, this brings up a massive 1990s era myth that Venki Ramakrishnan addresses, and that we really need to permanently debunk.
Oh, I know what you're going to say, the antioxidant craze.
Exactly. The old theory posited that aging was caused purely by oxidative damage from these reactive oxygen species tearing through our cells like shrapnel.
Right, so the logical assumption was that consuming massive, isolated doses of antioxidants, like huge vitamin E or vitamin C supplements, would neutralize the ROS, stop the damage, and therefore halt aging.
But the data didn't support that at all, did it?
No.
The clinical trials completely failed to show lifespan extension from mega-dosing antioxidants. And now we understand why. Reactive oxygen species are not just toxic exhaust. At certain physiological levels, they serve as vital communication tools.
Communication tools.
They are cellular signaling molecules.
Oh, so they aren't just exhaust, they're the smoke alarm.
Precisely. The ROS signal to the cell that it is under metabolic stress, which triggers the cell to activate its own internal defense and repair mechanisms.
So if you flood the human body with exogenous antioxidants,
You might extinguish the smoke alarm. You blunt the very signals your cells rely on to adapt, repair, and survive stress.
that makes perfect sense.
Bah.
So if the mitochondria are producing ROS as a signal, and the cleanup crews like autophagy are failing, what happens to all that misfolded protein garbage we talked about earlier?
It aggregates, it clumps together. We see this hallmark loss of proteostasis in several major neurodegenerative diseases.
Like Alzheimer's and Parkinson's.
Yes. But when we analyze studies testing experimental drugs designed to clear these protein aggregates out of the brain, we have to demand rigorous evidence. We must ask a fundamental question.
Which is?
Is the protein aggregate the underlying cause of the neurodegeneration? Or is it merely a downstream symptom of upstream metabolic failure?
Oh, like, is it the bullet or the blood?
Exactly. Some researchers even hypothesize the aggregates might be an active cellular defense mechanism, the brain attempting to quarantine toxic molecules by clumping them together.
Oh, so if a drug successfully clears the plaques out of the brain, but the patient's memory doesn't improve, the plaques weren't the root cause.
Exactly. What clinical endpoint was measured? Mechanistic claims require human clinical endpoints. Clearing cellular debris on a microscopic slide is completely irrelevant if it doesn't improve physical function, cognition, or survival in a human being.
That is such a good point. Let's follow that metabolic thread. If mitochondria are the power plants, how does the body decide when to run the plant at full capacity versus when to conserve energy?
This connects directly to nutrient sensing.
Right. Our cells have complex biochemical pathways with names like mTOR, AMPK, and insulin signaling. For a listener who isn't a biochemist, I think the best way to understand these pathways is to think of them as a combination of a fuel gauge and a general contractor.
Let's break down the functions of those specific pathways.
Okay, take mTOR, the mammalian target of rapamycin. It senses the presence of amino acids, basically protein, in the blood. If mTOR senses a lot of nutrients, it acts like a contractor who just got a massive delivery of bricks.
It wants to build.
Yes, it yells, "We have building materials, start constructing." It triggers cellular growth and reproduction. But if nutrients are scarce, mTOR quiets down.
And then AMPK, the fuel gauge, activates.
Right. AMPK senses low cellular energy. When it activates, it tells the cell resources are low, start building new structures, batten down the hatches, clean up the garbage, and repair the existing machinery. It turns off growth and turns on stress resistance and autophagy.
Which explains the biological mechanism behind calorie restriction.
Exactly. When scientists restrict the caloric intake of model organisms by, like, 20 or 30% without causing malnutrition, they are intentionally keeping mTOR quiet and keeping AMPK active. They are forcing the organism out of growth mode and into repair mode.
And in many laboratory models, this significantly extends lifespan.
It does.
But this is where we must subject biological mechanisms to human reality checks.
Hit me with the reality check.
Calorie restriction undeniably extends lifespan in many short-lived model organisms. But when evaluating claims that human beings should adopt extreme caloric restriction to live to 120, we have to ask critical questions. What organism was actually studied in the lab? Was it a microscopic roundworm, a fruit fly,
Right.
or a highly inbred strain of laboratory mouse kept in a pathogen-free bubble?
And how long do those animals normally live anyway?
Precisely. We also have to clarify the massive gap between average lifespan and maximum lifespan.
Okay, explain that difference.
Protecting a laboratory mouse from its normal environment, controlling its diet perfectly, and shielding it from predators might prevent early death from disease.
Which brings the average up.
Right, thereby increasing the average lifespan of that group of mice.
Right.
But it does not necessarily prove we have extended the absolute biological maximum lifespan of the species.
That distinction is huge. So when a listener sees a headline about a fasting diet increasing lifespan by 30%, they need to realize that was probably in a mouse that didn't have to deal with the stress of commuting, paying taxes, or fighting off a winter flu.
Furthermore, translating severe calorie restriction from a lab mouse to a human being involves serious risks.
What kind of risks?
While we do see certain metabolic markers improve in human trials of caloric restriction, it is not a benign intervention. In humans, severe long-term calorie restriction can cause dangerous nutritional deficiencies, profound loss of bone density, severe muscle wasting.
And muscle mass is so important as we age.
It is deadly to lose it as we age. It can also cause significant psychological harm and eating disorders. You cannot simply look at the mTOR pathway label and assume starvation is a safe path to longevity.
Okay, so if we can't safely starve ourselves into eternal repair mode, what happens when those repair modes inevitably fail? What happens to the cell when the DNA is damaged, the telomeres are short, the epigenetics are scrambled, and the protein garbage is just piling up?
The logical conclusion.
Logically, the cell should just undergo apoptosis-programmed cell death and clear itself out. But sometimes they don't. Sometimes they become zombie cells.
The clinical term is cellular senescence.
Right. When a cell accumulates catastrophic damage, it essentially arrests itself. It hits the brakes and permanently stops dividing. As you mentioned earlier with telomeres, this is actually a fantastic defense mechanism to protect the body against cancer.
We want damaged cells to stop replicating.
Exactly. The problem is they refuse to die quietly. They stick around in the tissues, taking up space, and they secrete a toxic cocktail of inflammatory molecules. They are essentially screaming, "I'm damaged," to the immune system.
And as we age, the immune system becomes less efficient at clearing these senescent cells away, so they accumulate.
And this accumulation poisons the surrounding environment. This contributes directly to stem cell exhaustion.
How so?
We rely on adult stem cells to divide and refresh our tissues, our skin, our blood, our gut lining. But stem cells need a healthy microenvironment, or niche, to function. If the tissue is packed with zombie cells secreting toxic inflammation, the niche degrades. The stem cells can no longer do their job, and the tissue loses its ability to heal.
The recognition of this process has launched an enormous pursuit of senolytics.
Drugs designed to hunt down and selectively kill these zombie cells.
Yes. In animal models, administering senolytics to clear out senescent cells has shown some improved functional outcomes. The mice appear healthier and more active.
That sounds amazing, though.
It does. However, as with every intervention we've discussed, we have to evaluate the evidence hierarchy rigorously. For humans, senolytics must be categorized firmly as promising but unproven and potentially hazardous.
Hazardous how? If the zombie cells are causing inflammation and exhausting our stem cells, why wouldn't we want a drug that wipes them all out?
Because of the evolutionary trade-offs we established in the beginning, we have to ask what comparison group was used in the animal studies, and we have to remember what senescence actually does. Senescence is not simply cellular waste, it is an active, vital biological response.
Wait, I thought they were just toxic.
Senescent cells play a crucial role in wound healing and, as you noted, tumor suppression.
They help heal wounds.
Yes. The inflammatory signals they secrete actually recruit immune cells to the site of an injury to begin the repair process. Ridding the human body of these cells completely could catastrophically alter our tissue repair mechanisms.
Yeah, wow.
You have to consider the whole organism. A systemic senolytic drug might successfully clear the zombie cells out of an aging joint, potentially reducing arthritis pain, but simultaneously impair your body's ability to heal a laceration. Or worse, remove the barrier that was stopping a microscopic tumor from turning into a malignant cancer.
That is wild. It's like firing the city's entire fire department because their big trucks constantly block traffic and cause congestion.
Yeah.
Only to realize you desperately need them when a kitchen fire actually starts.
Exactly. Biology is a delicate balance of risks.
And this localized inflammation from senescent cells scales up to a systemic level, doesn't it? Aging researchers call it inflammaging. This chronic, low-grade, sterile inflammation that just hums in the background of our bodies as we get older.
It's pervasive.
Yeah, and it's driven by senescent cells, accumulating tissue damage, and dysbiosis, which is the age-related breakdown of the healthy bacteria in our gut microbiome. This inflammaging drives almost every chronic disease of old age. But here's where my mind gets blown. When we look outside humanity to the broader animal kingdom, we see species that seem to completely bypass this structural decline.
Ah, the insights from comparative biology.
Right. We look at the naked mole rat. It's a rodent, but it lives for 30 years, which is insane for its size, and almost never gets cancer.
A fascinating creature.
Or we look at certain species of bats that live for decades despite having incredibly high metabolisms. Or the bowhead whale, which has millions of times more cells than we do, meaning millions of more chances for a cell to mutate into cancer, and yet they can live for over 2 centuries.
Truly remarkable.
They achieve this exceptional longevity through evolved packages of traits. Like the naked mole rat produces this highly complex hyaluronic acid that stops cancer cells from crowding together. The bowhead whale has incredibly robust DNA repair mechanisms.
They are magnificent examples of evolutionary engineering, but we must immediately address a major conceptual misconception that often arises when discussing them.
Okay.
The existence of exceptional animal lifespan extension does not guarantee human benefit.
But if we know how the bowhead whale repairs its DNA, why can't we just engineer that mechanism into humans?
Because biology is not a modular machine where you can swap out parts at will, we must push back aggressively on the idea of simple genetic swapping. You cannot use CRISPR to cut a longevity gene out of a bat or a whale and paste it into a human embryo, expecting a human to live to 200.
Why not? If the gene codes for a better repair protein, what does it help us?
Because these traits did not evolve in a vacuum. They evolved over tens of millions of years as highly integrated, interconnected packages tailored to very specific ecological contexts and life histories.
They're part of a larger system.
Exactly. A whale's DNA repair mechanism works in concert with 1,000 other metabolic adaptations unique to a massive deep-sea mammal. Dropping that single protein into human biology would likely cause catastrophic regulatory cascade failures.
Oh, I see.
Just as there is no single universal rejuvenating probiotic that will instantly fix human dysbiosis, there is no single gene swap that will replicate a naked mole rat's longevity in a human being. The interconnected complexity of human biology is infinitely more difficult to safely manipulate.
Okay, so genetic engineering with whale DNA is off the table. Let's look at the interventions that are currently on the table. Let's talk about the pharmacopoeia.
Let's do that.
Taking all this mechanistic knowledge we've discussed, MTOR, AMPK, mitochondrial function, protein clearing, what happens when we try to put those pathways into a pill. Because right now, the internet is flooded with experimental longevity protocols.
It is a booming market.
You have rapamycin, which specifically targets and inhibits that mTOR growth pathway. You have metformin, a cheap drug widely repurposed from diabetes treatment to target metabolic pathways like AMPK. And you have a multi-billion-dollar industry of NAD-related supplements and precursors designed to artificially boost mitochondrial energy production.
We need to be exceedingly clear and rigorous on this point, as this is where the most consumer harm occurs.
Right.
There is a dangerous misconception that if a drug is formally approved by regulatory bodies for a specific disease like metformin for type 2 diabetes or rapamycin for preventing organ rejection in transplant patients, it is therefore approved or inherently proven safe and effective for longevity in healthy individuals. It absolutely is not.
The logic jump people make is, well, the FDA says metformin is safe enough for millions of diabetics to take every day, so it must be completely harmless for a healthy 40-year-old biohacker to take to prevent aging.
Which is a fundamental misunderstanding of pharmacology and risk-benefit analysis. A drug that is beneficial when a metabolic system is entirely broken like diabetes can be actively harmful when introduced into a healthy, functioning system.
That makes a lot of sense.
Let's analyze the human evidence hierarchy for these compounds. Let us take metformin. The bold claims regarding its longevity benefits rely largely on observational signals in epidemiological data.
Meaning researchers looked back at medical records and noticed that diabetics taking metformin seem to live slightly longer or have lower cancer rates than diabetics not taking it, or sometimes even non-diabetics.
Correct. But observational studies detect associations. They do not prove causal effects. They are highly prone to selection bias, reverse causation, and healthy user bias.
What does healthy user bias mean in this context?
It means people taking metformin are interacting heavily with the healthcare system. They are seeing doctors regularly, having their blood tested, and often receiving counseling on diet and exercise.
So they're just generally taking better care of themselves.
Exactly. This completely confounds the data compared to someone who rarely sees a doctor. To state the facts clearly, metformin has not been proven in healthy population randomized, placebo-controlled clinical trials to extend human lifespan or delay aging.
What about rapamycin? Because that is the darling of the longevity community right now. It has consistently extended lifespan in mice, dogs, and other organisms by dampening that mTOR pathway.
Rapamycin does have an impressive track record in extending lifespan in several animal models. It is biologically fascinating. But we have to test the evidence in humans.
Okay, what happens in humans?
In human beings, rapamycin is a potent immunosuppressant. Chronic use can cause clinically important adverse effects, including dangerous suppression of the immune system, leaving the user vulnerable to severe infections, as well as causing significant metabolic disruptions like insulin resistance and elevated lipids.
Which is incredibly counterproductive if you're trying to stay healthy.
Precisely. These pharmaceutical interventions belong firmly in the promising but unproven category, and in many off-label uses, the entirely speculative category.
That's a strong statement.
I will state this as clearly and definitively as possible based on the current scientific consensus. No pharmaceutical intervention, no supplement, no peptide, and no biohacking protocol has established immortality, broad systemic reversal of human aging, or reliable extension of maximum human lifespan.
That is a harsh reality check.
It is necessary.
So, stripping away the speculative drugs, discarding the off-label prescriptions, ignoring the unproven supplements, and the futuristic gene therapies, what does the rigorous evidence actually support right now? What actually falls into the well-supported category for extending human health span today?
The well-supported findings are not glamorous. They will not generate billions of dollars in venture capital, nor will they make for viral social media content. But they are backed by the absolute highest quality of human epidemiological and clinical evidence.
I am bracing myself. It's the boring lifestyle advice we've heard our entire lives, isn't it?
It is the foundational reality of human biology. Regular physical activity, which naturally stimulates AMPK and maintains muscle mass and mitochondrial efficiency. Adequate nutrition without malnutrition. Restorative sleep, which is when the brain clears its protein aggregates.
Right.
Avoiding tobacco and excessive alcohol. keeping up with vaccinations to protect the naturally aging, vulnerable immune system, and diligently managing clinical metrics with your doctor, controlling blood pressure, managing lipid levels, and reducing metabolic risk.
It is incredible that with all our understanding of epigenetics and ribosomes, the best defense is still a brisk walk and a good night's sleep.
The basics work.
But I have a major pushback here, pivoting toward the end of Ramakrishnan's book.
Let's hear it.
Science is moving fast.
Yeah.
If we do eventually unlock a biological treatment, let's say 10 or 20 years from now, we develop a perfectly safe senolytic that clears zombie cells without impairing wound healing, or a targeted epigenetic reprogrammer that safely turns back the clock in our organs. What happens to society? Does this just become the ultimate luxury good for the ultra-wealthy?
That's the question.
Because we already know right now that factors like age-friendly housing, access to quality health care, environmental safety, and protection from ageism shape healthy aging far more than biology alone.
That is the crucial, unavoidable synthesis we must reach. Science can investigate the molecular mechanisms of aging with incredible precision, but biology cannot solve unequal later life outcomes.
So it just makes things worse.
If highly expensive, complex longevity interventions eventually work, they will undoubtedly immediately widen the existing, already vast health and wealth disparities in our global society.
Wow.
The billionaires who funded the research will access it first, while billions of others lack access to basic sanitation or blood pressure medication.
It creates a biological underclass.
Exactly. Furthermore, science simply cannot answer the profound ethical and philosophical questions this raises.
Like, what length of life is actually sufficient? Is 80 years enough? 100?
Yeah.
200? Or how does a society even function if nobody retires? If generation after generation is stacked on top of each other, all fighting for the same housing, the same jobs, and the same natural resources on a finite planet.
Precisely. Radically longer human lives will fundamentally disrupt every structure of modern society. It will break traditional models of retirement, caregiving, employment, and global resource use.
It changes everything.
These are population-level consequences that require broad, democratic, societal participation, not just decisions made in a biotech boardroom. The outcomes of the longevity quest depend entirely on how we structure our institutions and distribute health span, not just on counting the raw biological number of years lived.
Wow. Okay, to bring this massive journey all together, the science of aging is advancing at a breathtaking pace. We are finally understanding the incredibly complex mechanisms of our decline, from the translation errors in our ribosomes and the shortening of our telomeres, to epigenetic drift, mitochondrial exhaust, and the accumulation of senescent zombie cells.
It's an incredible time for biology.
It really is. But this knowledge is not yielding immortality. The responsible, scientifically grounded goal of all this research is to preserve physical function, compress the period of sickness at the end of life, and prevent chronic disease. We have to ruthlessly separate the alluring hope of complete rejuvenation from the hard, clinical evidence of human biology.
And as we conclude this analysis, I must formally state that this discussion is an independent COSMORA HQ book overview. It is not an official or complete audiobook. We preserve the complete canonical title and author, Why We Die: The New Science of Aging and the Quest for Immortality by Venki Ramakrishnan.
I highly recommend checking it out.
We highly encourage our listeners to access the full work lawfully through libraries, booksellers, publishers, or authorized digital and audio services to explore these concepts in greater depth. And please invite others to subscribe, comment, and share this episode.
If science can add years to life, how will we make sure it also adds life to those years?